Hello viewers, welcome to the CEC EduSet lecture. Today I will be speaking on the second part of the lecture, which is the karyotyping for chromosomal abnormalities. Now in this particular part I will be discussing about how exactly these chromosomal abnormalities occur, what is the phenomena under which these chromosomal abnormalities occur, and there are various syndromes which are there, and what are these syndromes which are associated with these chromosomal abnormalities. Now there are a number of chromosomal abnormalities which are there. So these chromosomal abnormalities have been categorized into two major classes. One is called as the numerical, the other one is called as the structural abnormalities. Now the numerical one are many a times referred as the aneuploidy, where there is a abnormal chromosomal number which is there, and this is due to the extra or the missing chromosome which is there. Now these aneuploidy are being occurring through the various syndromes. So one of them is called as the trisomy. So there are 3 copies of a chromosome which is there, and many a times there is monosomy also. So there is a single copy of the chromosome, of which the Down syndrome is one of the well-known example of the chromosomal abnormality. Beside that, there is trisomy 21, trisomy 13, trisomy 18, and the Turner syndrome, Klinefelter syndrome. So we will be discussing about various syndromes one by one. Now these chromosomal abnormalities are many a times result in the breakage and incorrect rejoining of the chromosomal segment. So there is a range of structural chromosomal abnormalities which result in the disease condition. Now how exactly and why exactly these chromosomal abnormalities, which can be numerical or the structural, occurs? So there is a phenomena which is called as the non-disjunction of the chromosomes. Now this is a basic phenomena, and we know that there is a technique, cell division methods, which is called as the mitosis and the meiosis technique. But what happens is, if this process is failed up in these particular things, so the chromosomes or the chromatid arms, they are unable to segregate normally into the daughter cells during either the meiosis or the mitosis technique. So these results in the change in the number of the chromosomes, so they can be trisomy or the monosomic condition. Now if the mitotic non-disjunction event happens during the early cleavage, the resulted individual will be a mosaic form with more than one cell line contributing to the phenotype, which is many times referred as the chromosomal mosaicism. Now if the meiotic non-disjunction can happen in either the first or the second division of this one, and the resulted gamete which can potentially produce either trisomic or monosomic offspring over here. So if we see over here this particular image, we can see in the first part of the image, the left hand side, it shows where the non-disjunction has occurred in the meiosis first, while in the second cases the non-disjunction has occurred in the meiosis 2. So if we can see over here in the meiosis 1, when there is non-disjunction occurring, both the chromosome, it moves to the daughter cell, and subsequently the sperms which are formed over there have got an abnormal type, and when it is fertilized with the eggs producing the gametes, so there won't be any normal gametes which will be formed. So there is a trisomic condition or a monosomic, where there is a missing chromatid arm which is there. Now if the non-disjunction has occurred in the meiosis 2, we can see over here this occurs during the gamete formation, and the gamete formation is also not properly formed. So there is a trisomic condition, monosomic condition which has formed. So by the development of these particular gametes, subsequently the child will be having an abnormal constitution of the cell structure or the cell chromosomal number within them. Now this is again another image which shows the normal meiotic process, while the non-disjunction which occurs in the meiosis 1 and the non-disjunction which occurs in the meiosis 2 process. So we can see over here in the normal type, all the gametes are having n number of copy number, while the non-disjunction in the meiosis 1, it is showing n plus 1 or n minus 1, while in the case of when the non-disjunction in the meiosis 2 takes place, it is either n plus 1, n minus 1 or n copy number over there. Now one of the first syndromes which we will discuss is called as the Pétaeus syndrome. So this is a very serious rare genetic disorder which is there. So there is an additional copy of chromosome 13 which is formed over there. So many a times it is referred as the trisomy 13. So a baby with the Pétaeus syndrome has got 3 copies of chromosome 13 instead of 2 copy number. Now generally 1 out of 5,000 births of the child, they are affected by the Pettau syndrome, and more than 9 out of 10 children born with the Pettau syndrome die in the first year of their developmental process. About 1 in 10 babies with less severe forms of the syndrome, they survive and they live more than 1 year. Now, the Pettau syndrome was first described by scientist Dr. Pettau in the year 1960s, and he found that it is mainly occurring because of the trisomy 13. So there is an additional 13th chromosome which is present, which is mostly commonly due to the Notch disjunction in the meiosis process, which more frequently occurs in the mothers of advanced ages. Now, the advanced maternal age is a very risk factor for the trisomy 13 due to increased frequency of the non-disjunction over here. Now, the child who is developing with this additional 13th chromosome or the trisomy 13 has got several defects, or there are several characteristic features, abnormalities within that particular child, like the cerebral defect. There is a apparent anophthalmia which is there. There is a cleft palate which is there, hair lip which is there, triggered thumb which is there, polydactyl which is there. So there are a number of syndromes the person or the child suffers from, and majorly there is a cerebral and the cardiovascular malfunctions which takes place within the surviving patient. Now if we see over here this particular image, it shows the child who is suffering from the Pettau syndrome, and then we can see over here there is a cleft, the hair lip cleft which is there. So the child also suffers from the heart defects. There is a brain and spinal cord abnormalities which is there, the poorly formed or the small developed eyes which are there, extra finger or the toes which are there. And if we see on the right hand side, there is a karyotype which is showing. So we can see there is trisomy over there. So we can see over here 3 chromosomes which are present over there. Now, apart from that, their lip or the cleft lip which is there, so the roof of the mouth which shows the cleft plate which is there. So the child suffers from low muscle tone also, kidney defects also, and the seizures also. Now the next syndrome which we will discuss is called as the Edwards syndrome, which is basically occurring due to the trisomy 18. So the trisomy 18 is also a rare but a serious condition over here. So the baby generally survives, but in the most of the cases it dies before or shortly after being born. Now the first Edwards syndrome was discovered also in the 1960s by the scientist. His name was Edwards over there. So it was a neonatal disease with a multiple congenital malfunctions and the cognitive deficit which were discovered. So this is a syndrome which is one of the autosomal trisomy syndrome which occurs, and it in the second frequency only to the trisomy 21 occurs. So there are various characteristic features which are associated with the Edwards syndrome over there, and after the baby is born, the child is likely to suffer from number of syndromes because of the Edwards syndrome or the trisomy 18. So there is a decreased muscle tone which is there, so hypotonia, the child suffers from low set of ears, internal organs forming or the functioning is not proper, mainly the heart and the lungs. There is intellectual or the cognitive disabilities which are there, which can be very severe also, overlapping fingers or the clubbed feet which are there, small physical size, head, mouth and jaw is there. There is a weak cry and minimum response to the sound the child suffers from. So there are a number of abnormalities which are there. Apart from that, there are severe syndromes also with the Edwards syndrome, like the congenital heart disease which is there, kidney disease is there, breathing abnormalities the child suffers from, mainly the respiratory failure, gastrointestinal tract problem, abnormality wall, hernia and many other diseases which are there. Now the next disease condition which I will be talking about is the Down syndrome, which is again occurring because of the trisomy of the autosome, which is trisomy 21. Now this Down syndrome was first described by the English physician John Down in the year 1866, but subsequently the chromosome 21 was established about 100 years later by Dr. Jerome in the Paris. Now the first presence of this copy of this chromosome 21 was discovered, and subsequently it was found that it was a trisomy 21 and most common chromosomal abnormality which occurs in the humans. Now it is found that most frequently occurring live-born aneuploidy is trisomy 21 that causes this disease. The incidences of Down syndrome, it increases with the maternal age, and it varies in different population. For example, 1 in 319 to 1 into 1000 live births it have been recorded. Now if we see over here the common traits which can be seen over here, so we can see the various abnormalities of this particular child which can be easily made out from the normal child also. Now in the Down syndrome, the child may be looking similar to the person, but there are various different abilities which are there. The person with the Down syndrome usually have a IQ which is a measure of the intelligence which is mildly to moderately low in the index, while the child or the person suffers from the speaking problem, so slower to speak than the adult child also is reported. Now along with that, there are some common physical features of the Down syndrome which are there, like the flat face which is there. Then there is a sharp eye which is there, a short neck, small ears are there, the tongue that tend to stick out of the mouth. Then there is tiny white spots on the iris, so colored part of the eye is there. Then the small hand and the feet are some of the common features of this particular syndrome condition. Now apart from that, there are certain other features also, like the poor muscle tone which is there, loose joints which is there. The children or the adults are shorter in heights than the normal person. Now the next disease condition which we will be studying is called as the Klinefelter syndrome, which is 2X and Y chromosome which is there. Now the Klinefelter syndrome is having one extra chromosome which is the X chromosome. So the copy number or the chromosome number is 47 in these particular individuals. So if we can see the karyotype over here, this karyotype shows the copy number where there is 2 X chromosomes which are there. So the chromosomal variation occurs in the males in which one extra X chromosome is there and is present. So the resulting phenotype or the karyotype is 47 chromosomes, while there is 2 X chromosome and 1 Y chromosome. So we can see over here in the karyotype all the autosomes have been arranged which have been characterized or characteristically stained with the dyes. So we can see the autosome along with the sex chromosome in which 2 X chromosome and 1 Y chromosome is there. So there is a chromosomal variation in the male because of the extra X chromosome which is present over there. Now this extra X chromosome, it typically affects the various functioning of the body parts, like majorly into the physical functioning, neurodevelopmental process, behavioral and the neurocognitive process. So the child or the adult suffers from various problems of the neurological problem, behavioral problem and the cognitive or the brain related or the various problems the child suffers from. Now over here, if we see some of the main or the key features of the Klinefelter syndrome which are shown over here. So majorly the male person or the patient is tall in height. He is sexually underdeveloped, so there is a infertility which is there because of the presence of a extra X chromosome. There is sparse facial or body hair which is there. Developmental process is delayed. There is increased risk of number of autoimmune disorders and breast cancer is there, or osteoporosis the person suffers from, leg ulcers, depression and dental problems which the person suffers from. So we can see over here there are a number of problems the person suffers for in the case of the Klinefelter syndrome. The next syndrome is called as the Turner syndrome. So the Turner syndrome, in the Turner syndrome, the female only genetic disorder, so it affects about 1 in 2000 baby girls over there. A baby girl with the Turner syndrome only has one normal X chromosome, so rather than the 2 X chromosomes. So one of the X chromosome has been missed out, or it has been missed out, or there is a non-disjunction due to which this has occurred. So the chromosomal variation, it occurs randomly when the baby is conceived in the womb. It is not linked to the mother's age. It has been found through the research. It is not linked to the mother's age over there. Now, the female with the Turner syndrome often suffers from a wide range of syndromes, and there are some distinctive characteristic features which are there, like for example the short stature which is there, lack of ovarian development, neck abnormalities which are there, skeleton disorders which are there, increased risk of osteoporosis, cardiovascular constriction, diabetes, kidney and thyroid problem. So if you see over here, in most of the syndromes, there is a abnormality development process and the growth which is taking place in the child or the adult stages. So these particular things are causing various problems in the subsequent ages, majorly in the cardiovascular problems, kidney problems, the locomotory problems and the physical problem, along with the various sexual problems also. So if we see over here, in the case of the Turner syndrome, there is lack of ovarian development which has taken place because of the missing X chromosome which is there. Now the Turner syndrome karyotype is shown over here. So we know that it is a karyotype of 45 chromosomes. So one of the X chromosome is missing over there. So many times it is referred as XO over here. So the female lacks the second X chromosome which is present in the normal karyotype. So if you see over here, this is a karyotype of the Turner syndrome where there is 45 chromosomes which are there. The autosomes are shown over there which have been arranged in the pair. So we can see the first, second, third, fourth, like this one, all the autosomes, 22 pairs of the autosome, along with the sex chromosome in which only one X chromosome is present while the other X chromosome is missing over there. So this forms the Turner syndrome where one of the X chromosome is missing, and due to this missing X chromosome, the female suffers from various disorders which we just discussed over there. And it also occurs through the process which we discussed in the previous slides, which is called as the non-disjunction. So the non-disjunction we discussed, it can occur in the meiosis 1 as well as in the meiosis 2 processes, due to which abnormal gametes are formed, in which the copy number of these chromosomes, they varies over there. Now the next syndrome which is there, which is called as the Fragile X syndrome. So it is also a genetic disorder which is there. So Fragile X syndrome, it, as the name is suggesting, there is some kind of abnormality which occurs in the X chromosome. So what happens over here in the Fragile X syndrome, it is caused due to or the changes in the gene which is called as the Fragile X messenger ribonuclear protein 1, which is called as the FMR1 over here. So the FMR1 there is some changes in this particular FMR1 gene, due to which the protein which is produced by this particular thing is not formed properly. So this protein is called as the FMRP over there, FMR protein, and it is one of the most important protein needed for the development of the brain processes. So the person suffer from many of the cognitive or the brain related problem. The people who have FX, which is the Fragile X syndrome, do not make this protein. So those with the Fragile X associated disorders have changes in their FMR1 gene, but usually still make some of the protein, but the person suffers from number of disease conditions over there. Now the FXS syndrome, which is the Fragile X syndrome condition, it affects both the male and the females over there. So the females have got a mild syndrome than the males over here. The exact number of the people who suffers from this FXS syndrome is quite unknown, but the recent studies of the researchers have estimated that about 1 in 7,000 males suffers from this Fragile X syndrome, while 1 in 11,000 females suffers with this FX syndrome over there. So we can understand over here one of the important protein which is not formed because of the changes in the gene which is there, due to which this protein is not formed and the proper brain functioning or the developmental process don't takes place. Now what are the signs that the child might have in the FXS syndrome is there is a developmental delays which is there. So the child is unable to sit, walk and talk properly at the same time, and the child suffers from various learning disabilities because majorly the brain developmental process is being affected over there. So the learning and the various skills are being affected over there. So this is one of the major problem or the signs of this particular Fragile X syndrome which is done over there. Now the social behavior problems also occurs in this particular child who is suffering from this particular disease condition, like the child is unable to make eye contact, suffers from anxiety, there is trouble in having proper attention for that particular thing, hand flapping, acting, speaking without thinking, and the child is not, it is being very active in these particular cases. So there is number of cases over there. So if we see the image over here, it shows the Fragile X syndrome gene which is being marked over there. So we can see there is a change in this particular location which is shown in yellow color over there. Now these particular karyotyping, they play a very important role in understanding the syndrome or the disease condition. So because these particular conditions, they helps the scientists to discover the various abnormalities, chromosomal abnormalities and the chromosomal aberrations which are taking place. So over here in this particular slide, I have shown up one of the latest techniques which have been developed, in which the chromosomes are being mapped with the help of the various fluorescent dyes. So this particular technique is called as the FISH, which is the fluorescent in situ hybridization technique, in which the probes are being mapped which are having the fluorescent dye of various colors. Now these fluorescent dyes, when they are being marked over there, they are hybridized with the chromosomes. Now they go and bind to the characteristic points in the chromosomes, and they bind over there, giving a proper colorations over there. Now each of the chromosome, if we see, analyze the image over here, each of these chromosomes, they are being shown with different colors. So these different colors, they gives a marked feature to these chromosomes, what exactly changes are into this particular thing. Along with that, we can see the karyotype which is there. In this particular karyotype, we can see all the chromosomes which are arranged over there, and subsequently the image of these metaphase plates or the karyotypes are taken, and they are arranged in a unified fashion, and these particular thing are called as the ideogram. So all these chromosomes in which the autosomes and the sex chromosomes are being arranged. So 1 to 22 pairs of these autosomes are arranged along with the sex chromosomes which are arranged over there. Now by looking at these particular structures, we can then tell whether there is a chromosomal structural change, means whether there is a deletion, inversion, translocation, or any kind of chromosomal change. Means what means that we will compare it with the normal chromosomes, and then we can check that how the patient's chromosome structural changes are being occurring over there, and these will help the scientists to analyze the disease condition or the genetic disease. Now if there is any change in the chromosomal number, that can be also visualized with these particular things. And if there is any chromosomal number change, for example the trisomy which we just discussed, that there will be a presence of extra chromosome, so extra autosome or extra sex chromosome which is present, we can easily visualize with the help of these karyotype, and then we can say that the patient or the child is suffering from these particular disease condition. And if there is a missing thing, that also we can analyze through these karyotypes or the ideogram. So these particular karyotypings are very important nowadays for the checking up and analysis and prognosis of the chromosomal abnormalities, and then subsequently it helps the doctor to analyze what kind of conditions or what kind of diseases the child is suffering from. Now if the child is suffering from various diseases, then the doctor goes from the treatment regimes, and there are various things which are available. Now there is a term which has come up nowadays which is called as the genetic counseling. So in this genetic counseling, the newly born child's parents, both father and mother, the chromosomal analysis is being done, and before the child birth, the chromosomal analysis is being done, and with the help of these analysis, the scientists or the doctors can say what kind of changes are occurring within the male or the female or the father and the mother type, and analyzing their chromosomal analysis, they can then subsequently make out what are the changes which will occur or what are the possible genetic diseases the inborn child can suffer from. So this particular karyotyping are very important nowadays for analyzing, depending upon so many large number of genetic disorders and the diseases conditions which are coming up. So these particular things are very important. Now with this particular thing, I've come to the end of my lecture. So in this particular part of the lecture, we have discussed about what are the chromosomal structural changes which occurs in the chromosomes. Firstly, we discuss about the chromosomes, how important they are, and how exactly the structural entity of the chromosomes are being formed, in which the DNA is wrapped onto the histones and how they form into the chromosome. Then we discussed about the process of the karyotyping, how the blood samples from the patient or the person is taken up, and with the help of the various treatment regime, the metaphase spreads are being made, and once we take the picture of that metaphase spread, we can easily make out what is the karyotype of that particular person, and that particular karyotype can be then analyzed or compared with the normal karyotype, and then we can say that what kind of disease condition the person is suffering from, and is there any disease condition the person is suffering from depending upon the karyotype. And these karyotype techniques we discussed are taking place with the help of the various staining techniques. Like there are various stains which are there, Giemsa stain, quinacridone stain which is there, and these stain techniques, how they help the person to analyze those particular things. And then we discuss the syndromes over here. So with this note, I come to the end of my lecture. Thank you very much for watching the lecture. Thank you.